In the world of science and engineering, there are many things to study. For example, mathematicians study what is. Physicists study what probably is. Engineers study what isn't yet. But the bioengineers, we study what never should have been. On this channel, that is very much the case. We've turned various vegetables into meat and are actively working on a system to play video games with a dish of living neurons, amongst other things. Though something we haven't done in a while is any real genetic engineering and messed with the life code of an organism. But that's easy enough to fix. Inside this box is my order for thousands of dollars worth of custom printed DNA, and today we're going to put each of them into some cells so I can show you what they do, and how they work. And there are some pieces in here that I have wanted for years. If this is your first time hearing of the concept of printing DNA, I am pleased to report that it is actually really old technology at this point, and surprisingly affordable. There are lots of companies now that'll print out almost any sequence you want, though of course the trick is knowing what sequence to order in the first place, but we'll get to that. There has been a slow and steady revolution in the world of biology that's taken place over the last 30 years, and what was once painfully expensive now is a service used even by plucky high school students. But what can we even do with a printed piece of DNA anyway? Why would somebody pay hundreds or thousands of dollars for a few micrograms of life spaghetti? Short answer, the limit is basically only your imagination. You can make an organism synthesize useful chemicals, or make rainbow fish, or custom flowers, living logic gates, or snozzberries that really taste like snozzberries. With enough time and resources, you could probably make most of the monsters from Dungeon Meshi. And while that would be unfathomably difficult, it's not actually impossible. It's simply a matter of skill, time, and having a nearly endless supply of money to throw at the printers. Over the years, I've had all kinds of weird and cool bits of DNA printed or sent to me, and it'll take a whole series to go through them all, especially because I keep buying or making more. In preparation for this video, I've been going through my collection and picking the best pieces, tidying up their designs, and having them reprinted. That way, they're as easy to use as possible and are nice enough to be worth selling to all of you. One of the nice things about DNA is that once you have the original sequence printed, it is much easier to make copies and share it. And for the last few months, we've been getting ready to add DNA, bacteria, and other biologicals to our store. There are a few places that sell DNA, but the ones that have the most selection and the highest quality are businesses that only sell to other businesses, or to academics and nonprofits. On top of that, the DNA they sell is designed for industry and academics. What we wanted to do is have a store that'll sell more broadly, and the DNA that we carry will instead be designed to be either an amazing learning experience for students, or things that are demonstrations of modern synthetic biology. And of course, there's a few things that we made because they're either just cool or a useful tool. Today, not only are we going to be learning how to code DNA and go through a lot of constructs to learn how they work and see the cool things they can do, many of them are available now with the plan to add more to our store regularly. And I have some truly magnificent things in the works that will be the subject of upcoming videos and are just finishing printing. So let's dive in and learn how to recode life itself. Like all coding languages, learning them is best done by starting with simple examples, and we'll build up to more complex use cases. We'll start with literally the most basic use case, making a bacteria produce one new protein, and we'll work up to building metabolic pathways, logic gates, and oscillators by the end of the video. In truth, though, there are actually a lot of ways to modify the genetics of an organism, and not all of them involve printing DNA. Before DNA printing was cheap and many genetic tools had been developed, scientists used things like atomic gardens to make their GMOs. Basically, the way this worked is that you have a specially built circular area where you can put plants, and in the center was a retractable pillar. Inside that pillar would be an extremely radioactive chunk of material that would allow you to irradiate all the plants you put around it. Once you'd irradiated them enough, you harvest their seeds and plant them and see if you get any weird plants that have new properties that you want. You then take those weird plants and breed them with normal crop strains to stabilize the gene. As you might imagine, there's nothing particularly efficient or fast about this process, but you've enjoyed the results of it even if you didn't realize it. For example, those red grapefruit you see everywhere were made this way. Normally, grapefruit didn't have that intense red color on the inside. But it's not just grapefruit, lots of crops were tweaked this way. Though for some reason, mutagenesis like this usually isn't considered genetically modified, even though you're changing literally thousands of genes randomly, for some reason this doesn't count. But mutagens are still used all the time to tweak and improve crops, and we use them in the lab as well. Even before nuclear gardens, humans have been messing with the genetics of pretty much every organism around us. For as long as there have been humans, there have been GMOs. My favorite example of this is Brassica oleracea. This simple plant started as field mustard, and it had a wide natural range. But everywhere it grew, different pockets of humans started growing it and selecting plants for characteristics they liked. Some people selected for terminal buds, which gave us cabbage. Some selected for lateral buds, which gave us Brussels sprouts. Some for stem, which gave us kohlrabi. Stem and flowers, and you get broccoli. Flowers alone, and you get cauliflower. Seeds, well, that's just mustard. And some truly deranged humans selected for leaves, and that's how we got stuck with kale. There are almost no foods that you have ever eaten that haven't been heavily modified by curious humans wanting to make their food just a little bit better. We just happen to exist at the end of a 10,000-year journey where most of the work has already been done. Corn was inedible, carrots were a joke, and almonds or tomatoes would probably have killed you. And it's not just the plants. Animals and even various fungi and microbes have been domesticated and tamed by human modification. Silkworms, bees, puppies, beer, chocolate, apples, potatoes, and turnips. All just as man-made as a Pop-Tart. Turnips, by the way, also brassica. But thankfully, we're not Neolithic humans anymore, and now we can just go in and directly change and add or remove whatever gene needs tweaking. So let's move on from random to precision, and learn how we can just print and insert DNA however we want. Now, the first thing you need to do before you start printing is pick the organism you want to work with. There are many to choose from, and scientists have also isolated examples from almost every area of nature and chosen examples to work with that have become so-called lab strains. Today we're going to be working with a bacteria you have almost certainly heard of before, E. coli. Now, if you've never worked with E. coli before, a brief word of warning. E. coli was isolated from a human stool sample nearly 100 years ago. As such, it's not so much that E. coli smells like poop, so much that poop smells like E. coli. In fact, around 25 to 55% of the dry weight of poop is actually bacteria, not the food you ate. So most of what comes out of you grew inside of you. Horrifying as that thought is. While E. coli is a fantastic organism to work with in principle, being easy to modify as well as being the most well-studied organism on Earth, the smell is not fun. That stank comes from a chemical called indole and is actually how the bacteria communicate with each other. Because bacteria have no mouths but must scream, they all produce small amounts of indole, but can measure the level of indole present in solution as well. When the level gets past a certain threshold, the bacteria know that their population has gotten too dense, and will slow down their metabolism to avoid depleting the food supply as quickly. They're also not the only organism that use scents to communicate like this. The smell of cut grass, for example, is actually grass screaming to their neighbors to warn them that something has been damaging them. And nearby leaves of grass will increase the amount of bitter compounds in response to discourage pests. Though unfortunately for the grass, that doesn't seem to deter the sharpened steel moving at many revolutions per minute. Anyway, all the DNA I'll be showing off today has been designed to work in E. coli, because as I said, it's a very easy organism to work with. For one, its doubling time is 20 minutes, so growing more is very quick. But beyond that, both its genetics and the physical makeup of its outer membrane make it particularly amenable to modification. E. coli in the wild have one main chromosome, but like many bacteria, they can also have extra pieces of DNA that float around called plasmids. These are small circular pieces of DNA that can be exchanged between cells to transfer new abilities if one of them evolves something new and useful. For example, the difference between dangerous E. coli and harmless E. coli is a plasmid that some of the cells carry that induces virulence and makes them dangerous. But without it, they're harmless, and some E. coli strains have been used as probiotic formulations. We can take advantage of all of this and design our own plasmid, and then stick that plasmid into the E. coli. If we design it correctly, it can't tell the difference between a natural one and our homemade one, so it'll run the program without issue. Think of all of this like installing new software. Instead of installing virulence genes, we're going to make the E. coli do fun and new things. Now, we're going to be designing our plasmids so that they continue to exist as plasmids. However, it's also possible to design them in such a way that the DNA will end up incorporated into the main chromosome, instead of just floating around on its own. But that's more complex, so we'll talk about that another time. For a plasmid to work, it really needs two things at a minimum: an origin of replication and a selection marker. Though that's just what it needs for it to exist. We also need to add whatever else it is that we want the E. coli to do, but those first two things are mandatory. An origin of replication is exactly what it sounds like. It's a place where the machinery inside the E. coli can grab onto the DNA and start the copying process. This way, as the cells grow and divide, new copies of the DNA will be inherited by all of the daughter cells. There are many ORIs to choose from, but generally they come in two flavors: high copy and low copy. This refers to how many copies of the DNA will end up being maintained in each cell. Low copy is in the 5 to 10 range, high copy is in the 400 to 600 range. So, just a small difference between the two. High copy is better for, well, making lots of copies of the DNA and physically producing a lot of DNA. Whereas low copy is better for protein expression because you're less likely to overwhelm the protein production machinery. With the ORI chosen, the other thing to pick is your selection marker. A selection marker is just something you can use to sort the modified cells from the unmodified ones. And since we can't go in with tiny tweezers and just pick out the good ones, this usually means something like an antibiotic resistance gene. This way you can kill off any cells that haven't taken in the new DNA by spreading the bacteria onto an antibiotic plate. When we go to actually put the DNA into the cells in a moment, only a tiny portion of the cells will actually successfully take it in. So we need to get rid of all the failures and allow only the modified ones to grow. Though, you don't need to use antibiotics necessarily. Of course, they're quick and easy, but that does mean you're spreading antibiotic resistance genes around. Other options can include things like synthesizing essential amino acids, and instead of antibiotic plates, you grow the bacteria on media that is missing that essential component. Or you give them the ability to eat something that normal bacteria can't, and then you feed it to them. Many of my plasmids are designed to work with an antibiotic called kanamycin. I like this one for two reasons. First, I'm allergic to penicillin family drugs, so I don't love working with ampicillin, which is what many other plasmids use. Secondly, because penicillin has been around for so long, many of the microbes in the air around humans actually tend to be resistant to it already. So you'll have much more of an issue with contamination with ampicillin than you will with kanamycin. Now, it may have sounded like you need to code all of this stuff up yourself, but in truth, this has all been done for you. There are many plasmids already available that come with an ORI and a selection marker already built in, as well as what's called a multiple cloning site, or MCS. The MCS is a spot where it's been purposely made very easy to pop the DNA open and slot in a new piece. This way, you don't have to print the whole plasmid each time, only a tiny little piece, and you choose what's called the vector or backbone according to your preferences and needs. Most of my plasmids are based around a vector called PET9A. It's a low-copy kanamycin carrying backbone, so all we need to do now is design the piece we slot in. So, with the basics sorted, what can we actually put in here? Well, let's start off easy. One of my favorite families of proteins are the fluorescent proteins. As their name implies, they're proteins that respond to UV light by glowing brightly, and they come in literally every color. They're also the closest thing to a Hello World equivalent that you're going to get with DNA, so are the perfect thing to start with. I am an avid collector of fluorescent proteins, and my freezer is packed full of different tubes of colors, and we actually did a video showing off the state of my collection a few years ago. But since then, I have added some truly incredible ones, and actually made a few of my own. Because these sorts of proteins are useful and variable, there's actually a whole website dedicated to cataloging fluorescent proteins called FPBase, where you can explore the different colors and find their sequences. However, it's not quite as simple as just copying the sequence you find on this site and pasting it into our plasmid. First, it needs to be translated from protein sequence to DNA, but that'll only give you the code for the protein itself, and pasting it into the plasmid won't have any effect. To make this work, we need two more things: a promoter and a terminator. Just quickly as a refresher, this is the central core tenet of biology. DNA is the information storage molecule, and proteins are the big important molecules that DNA usually codes for. But to get from DNA to protein, there's a middle step. The DNA first has to be read and a copy made out of RNA. It is this RNA, called messenger RNA, or mRNA, that is then read and transcribed into protein. And yes, mRNA is the same stuff used in vaccines. Because it will instantly induce the creation of protein as soon as it gets inside of a cell, but will then be quickly broken down, it makes a great vaccine platform. You can get a ton of protein produced quickly so your immune system can chew on it, but then the RNA is broken down and the process stops. Anyway, back to our code. Promoters are sequences where RNA polymerase can grab onto the DNA and start making that RNA copy. The RNA copy acts as instructions for the protein production factory that'll grab onto it called a ribosome. The ribosome reads the RNA in 3-letter chunks to know both where the start and end of the protein sequence is and what that sequence should be. But what happens when the ribosome gets to the end of the protein sequence? Well, there's a stop codon, but the other thing you need is called a terminator. This is a sequence that, among other things, forces the ribosome to pop off, and it will also make the RNA transcription stop as well during that step. This combination of promoter, protein sequence, and terminator is what's called an operon, and is the basic structure of all genes. So let's actually do it and try this out. To start with, I've got a plasmid that codes for a bright green fluorescent protein in this tube. But how do we get the DNA into the bacteria? Also, it's actually not enough to just say, I want to use E. coli, because there are many strains to choose from. I'm going to be using this plate of E. coli strain DH5A as the starting point for this process. It is a super common lab strain that both grows quickly and is easy to put DNA into, though it's not the best for protein production. For our purposes today, that's fine, and the ease of transformation is what we want, and you can get it now on our store. To get started, we need the bacteria to be very actively growing, so I'm going to set up a liquid culture to grow overnight. The liquid in this tube is called LB, or lysogeny broth. It's a mix of yeast extract and enzymatically digested milk protein called peptone, as well as a little bit of salt to taste. You can either buy this as a pre-mixed powder or buy the individual ingredients and mix it up yourself, which is what I do since it is much cheaper that way. We need it in 2 forms, liquid like this, and then also solidified into plates with the addition of some agar. For those plates, we need 2 sets, one with and one without our chosen antibiotic mixed in. Because E. coli aren't the only things that can eat LB, we need to sterilize all of this growth media, as well as work under sterile conditions to avoid contaminating our cultures or our workspace. Generally, sterilizing media is done in a pressure cooker of some kind, and I use these nice dental autoclaves for this. I'm mixing up multiple bottles with and without agar, but no antibiotics yet. These get added after sterilizing as they're heat sensitive. Then we load the bottles into the autoclave and let it cook for half an hour. When the cycle is done, the autoclave is carefully depressurized, and then the hot bottles can be removed and brought back to the lab. At this point, the agar will have melted, so while it's still molten, we can add the correct amount of antibiotic solution. Antibiotics usually come as a powder, so we have to make a concentrated stock solution, which are then sterilized with special syringe filters. These have pores that are so tiny that no living organism can fit through, effectively sterilizing any solution passing through one. Then when the bottles are cool enough that you aren't getting burned, but they are still very warm, we can pour the media into petri dishes which come pre-sterilized. These can then be allowed to cool and solidify, but with that done, we can finally prep our bacteria. By the way, I am breezing through this without giving much in the way of exact measurements, so there's going to be links in the description to written protocols for all this stuff for those that want to follow along. One of the greatest boons to modern biology is the overwhelming amount of information available online, from protocols to reagent info and more. And to be a biologist is to really collect and synthesize as much information as possible to create working protocols. Unlike chemistry, we can't really see any of what we're handling until the very end. It's all just moving small volumes of clear liquid around. The real magic happens by designing and correctly following protocols, and that's like 99% of the actual work. Alright, now with sterile growth media ready, I can inoculate 10 milliliters of normal LB with our E. coli. Then I'll pop it into my Tippy incubator at 37 degrees and leave it to grow overnight. You don't actually need one of these, a regular incubator works just fine, but these do make the bacteria grow a little better. The next day, I'm going to use 500 microliters of this solution to inoculate another 40 milliliters of LB. But this time, we're going to monitor it closely for a few hours. What we're waiting for is the bacteria to be in the middle of what's called log phase. When bacteria grow, they go through distinct phases. First, they're growing slowly for a while until a critical mass has accumulated and their doubling begins to make the population explode. At some point, when the nutrients start to run out, they slow down and hit the plateau phase, before eventually starting to die off. Log phase is when the population is exploding and the bacteria are at their most active. We can check to see where they're at by using a device called a spectrophotometer. It's basically just a light and a photodiode, and it'll measure how cloudy the solution has become by comparing between pure LB solution and our bacteria solution. This cloudiness is called the optical density, and we're looking for around an OD of 0.5. You can totally learn to eyeball this with enough practice, which is how I used to do this, but if you want the absolute best results, a speck is helpful. With our bacteria grown enough, we need to wash them to get rid of any of the LB and other salts or side products that will impede the DNA going into the cells. And to do that, we're going to be using a centrifuge and a bottle of sterile 0.1 molar calcium chloride solution. But before we do, I'm going to prepare a crushed ice bowl to do all the work in. From this point in the protocol, we want to keep everything very cold. Not frozen, but around 2 or 3 degrees. While the protocol will work without ice, we've checked, and ice actually improves the effectiveness enough to be worth it. Anyway, we take our E. coli solution and load it into our centrifuge with a matching dummy tube filled with the same amount of water on the opposite side to it. This way, the centrifuge won't explode. This will form a pellet of cells at the bottom of the tube. We can then carefully dump out the LB and replace it with 10 milliliters of calcium solution. Everything is mixed by pipetting up and down gently, then it's all spun down again. We're once again going to replace the solution, this time with 8.5 milliliters of calcium. Because the goal of this is to have frozen tubes for later use, we add in 1.5 milliliters of sterile glycerol on top, which will act as a cryoprotectant. Once that's mixed, we're going to aliquot the E. coli slurry 10 microliters at a time into a bunch of tubes. I'm using my fancy repeater pipette to make this fast and save on the carpal tunnel. These can then be quickly frozen in our -80 freezer and are now ready whenever you need them. If you want a protocol that doesn't make nearly as many tubes and only makes a single tube of bacteria, I've put a link to an older video where we go over it. All of these frozen tubes are what are called calcium competent. What we just did was strip the bacteria of its food, and with the cold calcium solution, we're decorating the outside of the cells with calcium ions, as well as opening tiny holes in the bacteria's membrane. This makes it so that any DNA we add in here will stick to the bacteria and have an easy time of getting into the cell. Speaking of which, I'm going to go ahead and do a bunch of tubes at once, since we went to the trouble of making a big batch, but each of the tubes will have a different piece of DNA added to them. To each of the tubes, I've added about 100 nanograms of their respective DNA. But if you're using a stubborn strain like BL21, you can increase the DNA concentration up to 1 microgram. And of course, be sure to carefully label each of the tubes, as it is impossible to know what these tubes are otherwise. Anyway, with our tubes of E. coli inoculated with the DNA, these can be put into the fridge for about 20 minutes to allow the DNA time to get friendly with the E. coli. Then we're going to do what's called heat shocking. Basically, we're going to stick the tubes into a heat block set to 42 degrees and let them cook for exactly 45 seconds. Then we plunge them back into ice. Basically, what we're doing is making the holes in the E. coli bigger and then in one big flush, forcing a bunch of DNA in and slamming the holes shut behind. To help them recover from this whole ordeal, we add 1 milliliter of LB and then let the tubes hang out in the incubator for an hour or two. Finally, 150 microliters of each solution can be spread out onto appropriate antibiotic plates. These are just left in the incubator to grow overnight. The next day, the plates should be covered in little colonies, and we can see what the DNA did. As a refresher, here is a regular plate of E. coli. Now, here's the one with the green protein. It already looks distinctly more yellow, but look what happens when I hit it with a UV light. It is so bright, you could comfortably read a book with this glow. Using a fluorescent protein like this is called using a reporter, because the glow is our signal, or report, that the modification worked. If we see it, we know the DNA must be present, so there we go, that's our hello world done. And this green, known as Fu-GFP, is easily my favorite. Okay, now that we've got the basics done, let's ramp up the complexity. As I said earlier, there are a lot of fluorescent proteins to choose from. Some don't even glow, but instead are brightly colored in white light. Probably my favorite of those is called Amyl CP. It's a gorgeous shade of blue, but it has always bothered me that you're forced to choose between something that either looks blue or glows blue, but never both. But, I mean, I'm a genetic engineer and I can print whatever I want, so I made this. I call it electric blue, and you'll see that it is both blue in white light and under UV. And the glow color really is a deep ultramarine blue, not the usual cyan of many other fluorescent proteins. To design this, all it really took was a little bit of copy and paste. I looked up the code for MLCP, and I found the code for a bright blue glowing protein on FP base called Electra. Then, instead of just pasting them side by side in the plasmid, I added a little flexible linker sequence between them. I also removed the stop codon from Electra to make sure that the ribosome reads through and makes this as one giant protein. The linker is made of a string of glycine and serines represented by these G's and S's. This will act like a big floppy noodle, but keep the two proteins connected, allowing them to do their things, but stay close together. Sure enough, it works exactly as designed, and sometimes designing proteins is literally this easy. You just find the bits of other proteins that do the thing you want, and you slap them together with a linker and express them as a fusion. Moving beyond electric blue, there is a whole collection of other weird fluorescent proteins. Another in my collection actually has the ability to change color. So it initially glows green, but when you hit it with a UV light, you'll start to see it very quickly start to change to orange. Now instead of responding to blue light and glowing green, it responds to green light and glows red. This is particularly helpful when tagging proteins inside of a big animal cell, because you can hit a specific spot with a UV laser, so only a tiny subset of the proteins in that cell change color. Then you can track each individual protein as it moves around the cell. In a similar vein, I have one fluorescent protein that behaves like a timer, and over the course of about 24 hours, it shifts its fluorescence from blue to red. Here you can see different cells seem to be glowing different colors, and you can see that the red ones are much older than the blue ones. It's also possible to build fluorescent proteins into sensors. For example, if you break one in half and fuse it to a protein called calmodulin, you'll end up with a protein that changes from dim to bright when calcium levels are high, which is the same sort of thing we used in the last episode of our neuron project to see individual neurons firing. When a neuron fires, there's a huge influx of calcium, making the protein glow brightly. If instead of bacteria, you modify something like a fish to contain this protein in its neurons, you can produce crazy videos like this where you see the whole brain firing. Though modifying animals is a slightly different toolset than modifying bacteria. Also, there's nothing to say that you should be limited to fluorescent proteins. You can express almost anything you want, though some things you'll run into some trouble physically or might be illegal. Toxins and infectious agents, for example, are almost always illegal without special licensing. But enzymes, on the other hand, are a great choice, and you can do a lot with them. Enzymes are just proteins that have a catalytic effect and will take some starting compound or compounds and modify them. And this is part of what makes biology amazing. With the right enzyme, you can stick all kinds of crazy molecules together and build it all up from basically sugar. Whereas trying to make the same molecule with classical chemistry could take a massive setup and horrifyingly toxic reagents. Here's a really fun example. ATF1, also known as acetyltransferase 1, is an enzyme that I took from baker's yeast. What it does is stick acetyl groups onto alcohols and forms what are called acetate esters. These compounds tend to smell fruity and pleasant and are part of why fresh baked bread smells so good. It's from the esters produced by the growing yeast as the dough ferments. Making esters like this is a classic high school chemistry experiment, but normally requires concentrated sulfuric acid and a reflux setup amongst other things. But by expressing the enzyme in E. coli, I can do the same reaction, but instantly and in a tube. If I inoculate a tube with the modified E. coli and then let it grow for a few hours, the E. coli will have produced a ton of the enzyme, and importantly, plenty of the special acetate reagent that's also needed, called acetyl-CoA. When I add a few drops of isoamyl alcohol to the tube and then give it a shake, within a few minutes the tube will start smelling pleasantly like banana as isoamyl acetate is formed. If we sub this out for other alcohols, we'll get other scents, like pear or citrus. Though the trick is to not let it grow too long, otherwise the butthole smell starts to become much more noticeable than the fruit smell. But we'll come back to this construct in the future as it's really fun to play with. But what happens if we want to express multiple enzymes? For example, in a previous video, we made yeast produce beta-carotene and turn the cells bright yellow and orange. But beta-carotene synthesis takes a bunch of steps, so we used a construct that had multiple operons. By following the pattern of promoter, gene, and terminator many times in the same plasmid, you can express multiple things and build pathways where chemicals are handed off between enzymes to make more complex chemicals, or break something big down. With beta-carotene, not only do you need the enzymes to make the actual carotene itself, you also need to add extra enzymes that make parts of the yeast metabolism more efficient and overproduce some of the starting materials. And this is the trouble you'll run into with pathways like this, balancing all the levels of everything and making sure that none of the chemistry is getting bottlenecked somewhere. Companies industrially do this all the time. There is a huge number of products that are made in either yeast or E. coli by expressing a few select enzymes to produce industrially useful chemicals. All those amino acids that get added to your sports supplement, for example, are grown in engineered organisms that have been tweaked to massively overproduce whatever amino acid is interesting. Now, Up to this point, we've basically just been telling the bacteria to crank out as much protein as possible because the promoter I've chosen is what's called constitutive, meaning it is always running. Constitutive promoters come in a range of strengths, so you can tune the levels of expression by adjusting the promoter, but that's a very permanent change in expression level and involves more DNA printing. What if we wanted to adjust the level on the fly? In nature, it would be very silly for genes to always be on all the time, even if they're not needed, as that would be very inefficient. So it should come as no surprise that every organism has many ways to turn genes on and off conditionally. One of the classic examples of this is the lac operon. One of the main sugars in milk is called lactose, better known as my ancient enemy. And while I did once genetically modify myself to be lactose tolerant, E. coli don't need to be, as they can eat lactose naturally. But because lactose isn't always present, being primarily in milk, the E. coli need a way to turn the lactose digesting genes off when not in use. This is accomplished with a protein called the lac repressor. It binds to a special sequence between the promoter and the start of the protein sequence and clogs up the machinery, preventing RNA polymerase from binding and the gene from running. But when lactose is present, it binds to the lac repressor, causing it to change shape and pop off, allowing gene expression. There are a lot of repressors like this that have been found and can conditionally turn a gene on or off. There are also the opposite, which are activators. These bind certain sequences and cause gene expression to increase by promoting RNA polymerase binding. This simple system is extremely important because in essence, what we've just stumbled onto is the biological equivalent of a transistor. Instead of controlling the flow of voltage, it controls the activation of a gene. Or with the repressors, it's more accurately a NOT gate. It inverts whatever signal it's given. Before we try wiring them together in bacteria, I've got a little setup made of actual electrical components to show you what I want to build and what we should expect. You can think of transistors like essentially just electrical valves. When you apply a little bit of voltage to their gate, they'll allow a lot of current to flow between the other two terminals. Here I have a pair of transistors wired up into what's called a bistable switch. Basically, whenever I press one of the two buttons, it will switch the state of the circuit and then stay like that. If I press button A, it switches to state 1, but if it's already in state 1 and I press the button again, nothing changes. But if I press button B now, it'll switch to state 2. If it's already in state 2 and I press button B, no change. In bacteria, we don't have switches and wires, and so we use repressors and the sequence they target to program what gets connected. And instead of switches, if we choose repressors that are sensitive to specific chemicals, we can apply those chemicals to cause the system to switch states. And that's exactly what I've built here. There are two sections, the part that acts like our transistors and the part that acts like LEDs. If we zoom in on the transistor part, we can see the trick to how this works. Normally, repressors are proteins that are always produced, and in turn they control gene expression by binding to specific chemicals. But what if we just stopped producing the repressor protein itself? We'd get the same effect, right? If there's no repressor present, there's nothing to stop gene expression. So what I've done is I've taken two repressor proteins, LAC and TET, but I've wired them to control each other. So whenever LAC is produced, it will turn off TET. If TET is produced, it'll turn off LAC. When a cell first starts expressing this system, it'll randomly settle into one of the two states. But we should be able to toggle between the two states by applying the chemicals those repressors respond to. To be able to see what state the system is in, I've put the same DNA binding sequences in front of two different colored fluorescent proteins, which will act as our LEDs. If the bacteria are red, they'll be in one state. If they're green, they're in the other. But we shouldn't see any bacteria that are yellow, meaning that they're producing both proteins at once. Sure enough, the initially transformed bacteria under the microscope show a very clear mix of red and green cells, but very few, if any, yellow. So the cells have correctly booted and settled into one of the two states. When we streak these out onto lactose-containing plates, we get red bacteria, and on anhydrous plates, we get yellow. Which means this is working perfectly. We made an actual living transistor circuit. Let's finish this video off with one last construct that is the sum of all we've learned. Once again, here is the electrical circuit I want to emulate. Basically, it's 3 transistors this time that are wired in a circuit. Each of the transistors can be imagined like a repressor. If they are arranged in a loop, this will induce oscillation as each transistor turns the next one off in turn. And we see the 3 LEDs blink in turn. Though this will only work with an odd number of transistors. If we have an even number, it's like the bistable switch and it'll settle into a stable state. But with an odd number, it'll always cycle. In this analog version, we see that the lights don't just turn on, they go through distinct brightening and dimming cycles as they turn on and off. And we should expect the same in the bacteria as well. The capacitors in this circuit sort of represent how much protein is built up. Even when the next element in the circuit is being triggered, it does take time for the previous one to clear. The DNA version is very similar. Like before, it's divided into two sections, the oscillator part and the reporter part. The oscillator is made of 3 repressors wired in a loop, and we've got 3 fluorescent proteins to act as our LEDs. And I've chosen the colors to be as far apart spectrally as possible to make them easy to differentiate and see if this is working. This oscillating construct is called the repressilator and was originally published in 2000 by Michael Elowitz and Stanislaw Liebler, as an example of an artificial circadian rhythm circuit. My homebrew version of the construct was designed to make it as easy to use and modify as possible, and hopefully look good on camera, so let's see how we did. Right off the bat, we see that different colonies are different colors, which is exactly what we want to see. In the original paper, their clock was so fast and consistent that they actually got concentric rings on their colonies. On mine, while there are a few colonies that had a bit of a ring structure, nothing like the original paper, which means that my clock must be running much slower. Under the microscope, we see all 3 colors, and the cells seem to only really express one color at a time, which is a really good sign. One of the cool tricks that the authors used were these special microfluidic flow cells that allow you to follow the lineage of a single bacteria as it grows, divides, and pushes the daughter cells out the little tubes. And you can see that the bacteria literally oscillate colors as they grow. Unfortunately, I don't have the special microfluidic system, and my microscope, while wonderful, is very clunky and shaky switching between the color channels. So I'm going to need to build a special rig to record a time-lapse of this properly, but that'll have to be for the future as it's a rather involved build. Even though I can't make a time-lapse of this yet, we can still test the rest of the circuit by streaking this out onto either lactose or TET plates like the bistable switch. And sure enough, it has a huge effect. On lactose, you get mostly red colonies with blue and yellow mixed in. And on TET, you get mostly blue with red and green mixed in. Under the microscope, we see the same. In the lactose version, we see lots and lots of red cells, some blue and some green. And in TET, we see lots of blue, some red, and some green. Unfortunately, the third repressor isn't triggerable, so I can't make it go mostly green. So the circuit definitely works. I think the tick rate is just really slow. This was actually something I was wondering about when I designed the circuit. Because a lot of the repressor proteins build up on each tick of the cycle, it takes a long time for it to be used up enough for the gene to run properly. It's like having a really large capacitor in the electrical circuit. It'll make everything tick super slowly, but it will still tick. The way the original authors solved this was by having a second sponge plasmid basically covered in binding sites for the repressors, on a plasmid that is high copy. This way, most of the protein that is produced gets purposely wasted, allowing the circuit to tick over much faster. I am thrilled that this worked, as it is the biggest genetic circuit like this I have ever coded up from scratch on my own, but I definitely want to make another accompanying plasmid or a second version with a built-in sponge. I'm releasing this first version as the base clock speed, and will release a faster clock in a bit. Also, this and all the rest of the constructs are designed to be easy to pop pieces out and replace them. Don't like one of the colors? No problem, it pops right out. Or you could apply this to something totally new and replace the colors with enzymes. The plasmids are all open source and you can change and use them however you want, and all the code is available on our GitHub. If there's a lot of demand for this and the rest of the constructs, we plan on releasing more plasmids regularly, as well as adding cool organisms to our store like our famous pet Jerry. Eventually, we'll also release kits for those that want to try their hand at all of this but don't have all the gear yet. Even though I think I just crammed the better part of an entire year of bioclasses into one video, I did have to skip over a lot of important bits. As such, I'm sure you still have a million questions, like how do you actually program this stuff on the computer? Or how do you order a construct from a DNA printing company? Well, thankfully you're in luck, because with this video, we just released a bonus companion video over on Nebula that covers at least some of those million questions. We go through how to use the DNA editing program you've seen throughout this video, as well as how you can find DNA sequences and how you edit your new monstrosity. Nebula, if you haven't heard of it before, is the amazing creator-owned streaming platform where all of your favorite creators have come together to make and share the content you love without having to worry about advertisers. And we are very proud to be part of the Nebula community. Not only are the videos completely ad-free, there is a ton of bonus content there too. This isn't the only video that came with a Nebula companion. Our Egyptian mummy project had one too, going over all the spells, hieroglyphs, and recipes used in that video. Beyond those Nebula exclusives, there are also Nebula originals, like Mustard's The Origin of Stealth, exploring the engineering of the F-117 Nighthawk, one of the coolest looking stealth aircraft. And if that wasn't enough, there are also Nebula classes where creators teach you about a wide range of topics. One that is particularly relevant today is How to Research Like a PhD Student by Tom Nicholas. It's a great dive into how to prepare for a project like the ones you've seen today. Nebula is the fastest growing independent video platform, and we are joined by so many amazing creators like Practical Engineering, The Hacksmith, Becky Stern, Lewis Wise, and too many others to possibly list them all here. Nebula is also helping creators like us elevate our content and take it to the next level. Better production value, bigger stories, all without having to chase the trending tab or appease the whims of advertisers. So if that sounds good, then use the link in the description to get a year's subscription for the incredibly low price of $36, or just $3 per month. Or the best deal of them all, which is a lifetime subscription for only $300. I can't think of any other streaming platform that is as good value for the price. So head on over to nebula.tv/thethoughtemporium to sign up today and upgrade your viewing experience. That's all for now, and we'll see you next time.